So these particles seem to be large, long fibers. So what we'll do is we'll go over the data. So I know there were two reports that were generated. There were two samples that were generated. We're going to go over some of the features of the particle insight, and then we'll also go over some of the comparisons and things that we can get from the particle insight. So the first thing I usually do whenever I start a brand new analysis is I will actually go in and look at the actual, just the basic thumbnails. But before I do that, what I'd like to do is I'd like to give you an overview of a couple things. One thing is there is a very good document that we have which goes over all the different shape parameters of the particle insight. It goes over, why is shape important. And it'll also go through the different shape measures as well. So the different shape measures will show you, circular measures, rectangular measures, and not all of them always apply. So for example, in the case of these samples that we were running here, we'll notice that, assuming particles are spherical or round is really not going to give you a whole lot of information. So if we were to run these samples in a particle size only analyzer that will give size only data, you're really not going to be getting a full view of what your particles are really like as far as analysis. The other thing I like to show customers is why so many different shape parameters. We have many different shape parameters. At the end of running these 34,000 particles, we end up with up to 32 different histograms. What I do is I try to show them this information. This is just a template where I can show that there are parameters that are generated that are size independent. I mean, these are being blocked off because there's some rejections, but assuming particles are spherical may be acceptable if they're round, but if they're not round and they're long fibers, such as this case, for example, in this case, this fiber is coming out to be in a size only measurement. So ECA diameter, equivalent circular area diameter, assuming the particle is round. It would assume the particle that would be 13 microns, which is obviously very incorrect. So in cases like that, we try to use some other measures. For example, in the case of these samples that we're running today, I looked at both bounding rectangle aspect ratio and Fourier aspect ratio, which is a microscopy type of caliper dimension. So I think those are a little bit more accurate in the samples that we were looking at. So going into the samples that we're looking at, I just had one of the measures here. Let's open some of the ones that I think were more accurate, which would be let's say the Fourier, let's say length. Let's look at the actual width of the particles, and then let's look at the actual aspect ratio of the particles. So what we have here is basically the aspect ratio of the particles. We went all the way out to 100, so there is not a whole lot of resolution here, but we'll be able to adjust that if we want to. But we can see that there's obviously a distribution of different widths of these particles, and there is a interesting distribution of different lengths of these particles. So by looking at this, it's interesting to see that the different lengths, some could be pretty long, and the width, some could be pretty wide actually. So that's pretty interesting. So what we'll do is look at these histograms. Very interesting information. Not a whole lot you can get out of it. Where we start looking at data results, especially when we finish an analysis, is we'll look at the actual thumbnail images. When I look at the thumbnail images, it's a general view of every single particle that was analyzed. So what I'll do is I'll look at the thumbnail images, and this is always something interesting to see. It's just a general view of all 34,000 images. So this is more qualitative than quantitative, to be honest with you. When we look at all the thumbnail images, this will give you a general idea of what these particles look like. However, it's kind of difficult to actually go and sort through all 30,000 particles. I can start scrolling through and go through all 30,000 particles, which would take me forever. But again, this gives me qualitative information. It tells me if there's agglomerates, if there is any large contaminants, if there's, I'll look at the different population, and I'll be able to see just different overall images that I'm looking at. So obviously I have dark and light stuff. I have thin stuff and some longer thin stuff, wider stuff. So there's a whole slew of information. But so right now we've gone through all the images. We have 30 different shape measures. What do we do with that? So this is obviously just a lot of information. So what you do with it is actually what's really of value. So it's not as how much information you have, it's what you can do with it. So when I start looking at some quantification of information, that's where I start getting some more value out of the data. So looking at, for example, here's one. I use this quite a bit. This is the correlation plot. This is a rare event detector. So what I will do with correlation is I will look for any out of the ordinary things. So one of the things I'll look at is, I tried doing this earlier, I'll look at the, let's say the Fourier aspect ratio width. Let's say look at the width of the particles compared to the length of the particles. So I don't know what these particles are. I'll just take a guess. And if I look at a comparison between the width and the length, maybe I can get some information out of what I'm seeing here. These little lines are kind of interesting, but generally what I'm seeing here is that it's somewhat linear, where as the particles get longer, they're getting wider in a linear fashion. So they'll go up to about a length of about 50 microns, but they're only going to go to a width of about 30 microns. But it looks like whatever process is being used to actually make these fibers, it's fairly uniform all the way down. And generally, when I see some of these stratifications in some of these correlation plots, it usually tells me a little bit more about what the process is going through. But you can see it's very, very linear, and there's no rare events outside of the norm. You can compare any two shape measures. Trying to think of what else could be a good. Diameter is really not a valid shape measure, I think, for this, because you're assuming it's sphere. Circularity doesn't really, maybe smoothness, maybe looking at the smoothness of the particles compared to the length. That really doesn't tell me a whole lot. But anyway, comparing any two shape measures to each other is a good way for looking at rare events and identifying what some of the strange things. And then obviously we can go in and look at the actual thumbnails of what those are and give it an attributable cause. Another very valuable tool to actually give you quantifiable information is the particle classification file. So the particle classification feature. So the particle classification feature is very unique in that it allows you to look at subcomponents of the population. So I created this a few minutes ago, and what I did is I said, okay, well, let's assume that there are two populations in the sample. Let's assume there are some wide particles and some long but thin particles. So I set up some parameters on what I would consider a long wide and a long thin. And what I can do is actually I can do an individual analysis. So not only can I tell you exactly how many we found, and I can tell you what the concentration of this was, but I can go in and look at just the statistics. For example, I can look at just the statistics of the long wide samples. So now, looking at the long wide samples, I think I chose something like aspect ratio, or did I choose? I think I did choose aspect ratio. So I chose aspect ratio to eliminate the ones that were long and thin. So this would only be the long wide samples, eliminating all the long thin samples. So I can undo that. I can go back and I could actually say only show me the long thin samples, or besides the statistics of just the long thin or just the long wide, I can actually just show the thumbnails of the long thin or the long wide samples. So obviously my parameters were not that great because these are they're short fibers, but they're actually pretty wide. So I think what I did is I chose an aspect ratio that was pretty short. And that makes sense. By having a lower aspect ratio, I thought they were going to be shorter, but they're obviously that doesn't necessarily mean the case. It could be long, but they could be wide. So it's just by aspect ratio where I limited it. So there's different parameters that you can choose in the particle classification feature to actually do any kind of subcomponent analysis. Again, this is another feature that's really useful for extracting more information. So what we can do now, just to see if we have a comparison, so there were two samples that were sent in. We can actually do a comparison of the two. Let's compare the two and do what's called a multiple run summary. So if we do a multiple run summary, what we'll do is we're going to actually compare these two samples that were analyzed, and then we can look at, so if we had a size only instrument, we can actually do a size only comparison. But again, I don't think that's a very valid point, because assuming these two particles are very similar, and as far as long and fibrous and long crystals, looking at just ECA diameter is really not going to be a good way of evaluating the differences. What I'll do is I'll look at things like, let's see, circularity, elliptical width. Again, these are all going to be in that the PDF I'll send. But let's go back and look at Fourier width. Let's look at the width of the particles compared to each other. So this is the mean width, and it's by number. And we can see that, okay, so it looks like the blue sample, which is the second sample, which is the number 2, it had some slightly wider particles. So it has a slightly wider particles. There are some small particles here, but definitely some slightly wider particles. This is on a number-based distribution. You could look at it on a volume-weighted distribution. No, we'll give you the same thing, because this is a width measurements by number. So we could look at width. We could look at length. So how does the length for a length compare? So again, the blue one, the second run here, this 284-2, which we had not opened. We were looking at the 283-1. It's also longer. So it is wider and it is longer. It could very well mean that my aspect ratio might be a pretty good overlay compared to each other, and it is. And that's because it looks like, since the second sample is wider and longer, the aspect ratio would probably be the same. So an overlay and aspect ratio would be very similar, which is what we're seeing. I'm just curious now. I want to go open in the second file, because I hadn't seen those yet. So these were longer and wider. Just out of curiosity, I'm just interested in seeing some of the thumbnail images and see what they look like. So let me open up the post-run processing, and I will look at the thumbnail images to see what the second sample looked like. Okay, so here's the thumbnail images. Wow, big particles. But let's see. Slightly out of focus. I think we probably could have improved the focus on these a little bit better. Anyway, so very interesting. Again, if I knew what these samples were, maybe these particles would be of interest. We can actually determine what population of blocky stuff there is compared to fiber particles. That's something we can do. We can do different comparisons. These are definitely a bit out of focus, but still gave us some pretty decent information. But anyway, that's pretty much it. So again, very good amount of information that you get. It's what you do with it. Looking at the thumbnails gives you some kind of a qualitative measurement, but then looking at correlation plots, classification, overlays, and you can do a lot of different Excel exports as well. It gives you a lot more information to make it more quantitative. I hope this helps. If there's any questions, please let me know. Thank you.